Hey, good afternoon, guys. Let's go over chapter 20 here on the pathogenic gram-negative cocci and bacilli. Recall that pathogenic refers to microorganisms that cause disease in humans, and cocci and bacilli are the shapes of the bacteria. So cocci referring to a spherical shape, and bacilli referring to a rod shape. So some of the most important genuses that out of which I'm gonna go ahead and cover some of the details on a few of these are Neisseria, Enterobacteriaceae, Pasteurellacea, kind of a mouthful there, Bartonella, Brucella, Bordetella, Burkholderia, Pseudomonas francisiella, Legionella, Coxiella, Bacteroides, and Prevotella. Again, these are some of the most important genuses, out of which I'll be covering, some of the detail of at least half of them. So let's go ahead and get started. So some of the most important characteristics of the gram-negatives would be, the fact that they all release a toxin called lipid A. That's one, and two. From the gram stain they're gram-negatives, therefore, that they're going to go ahead and stain either pinkish or pinkish to reddish, and you'll be able to see that under a light microscope. So some important things that you should know regarding all of these gram-negative bacteria, so in general, is that first is they constitute one of the largest groups of human bacterial pathogens. Okay, these are bacteria that live in and on us, most of these, and they don't necessarily cause disease most of the time, but when they do, right, they will be pathogens, and given the fact that they do become pathogens, that means that there was an infection going on, right? So they like to grow in cultures, so if you do cultivate them, they will grow at around 37 degrees Celsius. Keep in mind that our body is, right, the normal temperature of our body is about 37 degrees Celsius, which is 98.6 degrees Fahrenheit. So these bacteria, right, they do grow under this type of temperature. Therefore, our body is the perfect host for these for the culturing of these bacteria. So, what makes them such excellent human pathogens? Well, first is the fact that they do again release lipid A, which is an endotoxin. Right. So this lipid A is going to trigger many different symptoms, starting off with fever, which is an increase in systemic body temperature, vasodilation, which means that the blood vessels arteries for the most part are going to go ahead and dilate. They're going to go ahead and open up, therefore probably causing a decrease in blood pressure, right? So if you open up the arteries, then you decrease peripheral resistance, which decreases blood pressure. They're also going to cause inflammation, right, with all of their cardinal symptoms of the inflammatory response. For example, edema, redness, localized increases in temperature. They can also cause shock because of the very low blood pressure. In case a patient does suffer from systemic and sustained vasodilation, then that can pretty much bring them into shock. And last but not least is disseminated intravascular coagulation. Disseminated meaning systemic, right? So spread out throughout the body, intravascular, inside of the blood vessels. And the coagulation is simply the aggregation of these pieces of megakaryocytes called platelets, right? So platelets are part of our immune system. So their aggregation causes coagulation. That's part of hemostasis, right? which helps us in basically healing, right? Healing wounds to blood vessels. Again, they are gram-negative bacteria, so therefore they do stain pink or red. If they do manage to breach the skin, the first line of defense, then they're going to go ahead and cause disease. If they can also breach the mucous membranes, and they can also cause disease. Again, they grow at 37 degrees Celsius, and they can suppress the immune system of a patient. So these are the pathways in which lipid A, released from gram-negative bacteria, induces fever, intravascular coagulation, inflammation, and shock, right? Let's go over some of these, because these are very, very, very important mechanisms here. First is you have, again, the activation of leukocytes, the release of leukocytes and cytokines, right? What these are going to do is, these are chemotactic molecules. These cytokines right here, like interleukin 1, interleukin 6, interleukin 8, these are chemotactic molecules, meaning that they are signaling molecules that signal the immune system cells to come out and do their job, right? So normally, you would think the release of these would be good for the immune system, but keep in mind, this is lipid A. This is something foreign causing their release, right? So if we do release them in high quantities in a very short period of time, then they're probably going to tend to be immature, right? So these cells that are caused by the release of chemotactic molecules like IL-1, IL-6, and IL-8, so they're gonna be immature. They're not gonna have time to go through normal development, and therefore, at the same time, they're gonna go ahead and cause damage to blood vessels. How does that happen? Well, due to the fact that these cells, like neutrophils, for example, they act on the blood. They have an effect on the blood and blood pressure. What they do is they are released as a consequence of damage to the blood vessel walls, right? The endothelial layers of the blood vessels, if they become damaged, or if there's systemic inflammation going on, then they start undergoing processes like diapedesis, for example, and margination, which allows these cells to penetrate and percolate through the layers of the blood vessels, right? So they percolate through, and these, keep in mind, these are immature cells that are being released, and these are chemotactic molecules that are constantly being stimulated by lipid A endotoxin, right, from the gram-negative bacteria. So it acts as a cycle there that just continues to open up these blood vessels due to margination and diapedesis processes. So ultimately, if that happens chronically, right, so these blood vessels are going to become damaged, leading to a subsequent reduction of blood pressure. Now, let me get into more specifics on how blood pressure is reduced there. So given the fact that there is either systemic or localized inflammation, so that leads to vasodilation, right? So blood vessels tend to dilate, and that is to increase localized blood flow to specific areas of the body. So when that happens, right, and if that happens systemically, then you're gonna have an opening of the blood vessels throughout, or the arteries throughout the body, which again, as mentioned earlier, that causes a decrease in peripheral resistance, right? Again, keep in mind, I'm talking systemically here, right? So if it does happen throughout the entire body, then peripheral resistance is going to decrease, which is going to make it easier for blood to flow through those arteries and veins, therefore reducing blood pressure, because the heart doesn't have to work harder or hard enough in order to push that blood through. So again, systemic blood pressure reduction is a consequence of the release of immature neutrophils and also the release of chemotactic molecules like interleukins. So other cells that are stimulated by the release of cytokines are called tumor necrosis factors, or TNF, right? So these molecules, what they're going to do is they're going to be involved in fighting against the formation of tumor cells, but they're also released as a consequence of inflammation, and also released as a consequence of viral infections and bacterial infections. So, their release causes the onset of fever, right? So their release causes a pyrogenic increase throughout the body. The hypothalamus resets essentially the thermostat, if you will, increasing the temperature, releasing pyrogens to also increase body temperature, and that we know of as fever. Here's another mechanism, another route of or mechanism of action of lipid A, endotoxin. It's the activation of platelet factors. So platelet activating factors are basically stimulated, right, by the release of lipid A, which causes a release of more platelets, right, an increased release of platelets from megakaryocytes. So therefore, blood clotting reactions set in, and again, if that happens systemically, right, that can be disseminated throughout the body, causing intravascular coagulation throughout the entire body, right? So throughout the body. Why is that bad? Well, because again, these are blood clots that we're forming here, right? So normally you would want to form blood clots in cases of injury to blood vessels, right? So those blood clots basically seal off the injury, right, the wound. But then that process is called hemostasis. At the end of hemostasis, we have to get rid of that clot, right? So once we have sealed off the injury site, we get rid of that clot, of those clots, by using, things like fibrinogen and fibrin factors. So we dissolve those clots. Now, if we have disseminated coagulation, then the rate at which those clots are dissolved is not necessarily going to be optimal. Therefore, you might, or the patient might end up with clots everywhere, right, throughout their body, which can be problematic as well. And last but not least is the activation of complement proteins, right? Keep in mind, the complement system is a set of about 30 proteins, give or take, which are denominated C1 through C9, all of which are divided up into A, B, sometimes C. So what they do, right, the complement system, is it plays a role in inflammation, and also in activating some opsonization processes and neutralization processes that I talked about back in the immunity. So if that's activated right there by lipid A endotoxin, then inflammation sets in, which, if coupled with DIC and fever and systemic blood pressure reduction, then the person will for sure go into shock. And shock is characterized by first and foremost loss of blood pressure. That's one. Second is highly reduced tissue perfusion, meaning that the tissues are not going to get enough nutrients, oxygen, right, because of the loss of blood pressure. Dangerously low heart rate. So anything below 60 is a low heart rate or bradycardia. So patients in shock can go all the way down to the 30s, right? Which is dangerously low heart rate. And evidently, tissue perfusion goes down, which deprives the tissues of their nutrients, their oxygen, and so on. All right, so let's go into our first genus, gram-negative genus. This one's called Neisseria. So this is the genus Neisseria, and the disease I'm showing here, or the infection I'm showing, is Neisseria gonorrhea. So the specimens from the Neisseria genus, right, would cause disease in humans very regularly. So these are aerobic, non-motile gram-negatives that are basically arranged in a diplo cocci arrangement. So what does that mean? Well, that means that they are spherical and they come in pairs for the most part, right? So you see the pairs right there, pairs. And then they're aerobic, right? Meaning that they need oxygen to be able to carry on with their metabolism and survive, right? So they can survive in an oxygenic environment. They're non-motile, meaning they don't move, they're stationary, but they do grow in the anatomical location that they happen to be infecting. Right? Now, the next characteristic here, at least biochemical characteristic in terms of their testing, is that they're oxidase positive. So if after the gram stain you carry out so the biochemical testing in order to be able to identify, right, what the bacteria is in a sample, then if you use the oxidase test, then it's going to come out positive, right? So this distinguishes this bacterium from other gram-negative pathogens. So the pathogenic strains of Neisseria gonorrhea, they will contain Fimbriae, right? They will have a polysaccharide capsule and a cell wall that contains Lipidae. Again, Lipidae would act as an endotoxin to our tissues, right? So once we become infected with it, if we do become infected with it, and Lipidae is a molecule that is embedded on the membrane, right? on the cell wall rather, of the bacterium, right? So it would be found all throughout, all around the cell wall of the bacteria there. So there's two species of importance to us, again, one of which I'm showing you right now, Neisseria gonorrhea, and the other one is Neisseria meningitidis, which causes the disease bacterial meningitis. You most likely recognize the disease that Neisseria gonorrhea causes. It's evidently called gonorrhea. It is a sexually transmitted disease that occurs only in humans. So we're looking at the incidence, right, of this disease per 100,000 members of the population, and then the years. So look at the 1945 to 1950 range there. It did show an increase, right, and a pretty high incidence related to anything after the 2000s. And then look at the 80s, 1975 to about 1985. Look at the high rise, right, of the incidences of gonorrhea in the United States, and that obviously in relation to what is happening after the 2000s. Could it have to do with the availability of antibiotics, right, and treatments for this disease? Well, probably, right? So again, it is a disease that occurs only in humans, and it is sexually transmitted And there is an increased risk of infection with increasing sexual encounters. Yeah. So most cases in the U.S. are going to occur in adolescence, at least in dealing with the statistics. And it is pretty evident from the graph here that cases have declined over the past year. So after the 2000s, actually after the 90s, right, cases started to decline significantly, and so on. So as it does adhere to the genital tracts, urinary tracts, and digestive tracts. So given the fact that it is a sexually transmitted disease, right, condoms can do so much, right? They can only do so much. But in the case that there is an exchange of sexual fluids, right, genital fluids, then it is very possible that droplets can contain hundreds or even thousands of the bacterium, and that would basically constitute an infection on whether it's transmitted from female to male or from male to female. And there, you don't need a lot of copies of the bacterium, right, to be able to get infected with at least one droplet. That would be enough. It's just one bacterium is more than enough, right, for you to be infected. One bacterium that adheres to the urogenital tract of the male or the female, and this bacterium adheres well enough that they manage to begin asexual reproduction, right? And it's simple because they divide by binary fission. And aside from lipid A, endotoxin, other reasons why the bacteria is so successful inside the body is twofold, right? First is they do secrete protease that interacts with immunoglobulin A, which is IgA, which is an antibody, and it cleaves that antibody, right? So it goes ahead and cuts that antibody and then destroys the antibody. So if there's no antibody against Neisseria, then, or a low count of antibody against Neisseria, then it's going to be hard for the body to fight it off. And the second reason why they're successful is because they do survive within neutrophils and act as intracellular pathogens. Yeah, so neutrophils are phagocytes that go around the tissues looking for bacteria to engulf. So if these bacteria manage to enter neutrophils, then can the neutrophils engulf them and eat them? Well, not necessarily. So some of the symptoms that gonorrhea causes in men would be inflammation, which causes painful urination and pus-filled discharge. Again, the bacteria does adhere to the urogenital tract, therefore, triggering inflammation, right, in that system, which is the source of the pain that originates from urinating. The pus-filled discharge is due to the fact that pus essentially contains immune system cells, right? So if immune system cells collect around the urogenital tract, as you're pushing out urine, then pus is being pushed out as well, because pus will again fill the urogenital tract, and urine, as it passes down the urethra from the bladder, does push down pus, which causes pus discharge through the urethra. It often goes asymptomatic in females, right? So causing no symptoms, but if left untreated, right, which is very common if the disease goes asymptomatic, then it will trigger a more pronounced symptom in females called pelvic inflammatory disease, or PID. How does this happen? Well, the fact that the bacteria is not causing any symptoms, right, and that's not prompting, right, the female to act against it. It is not prompting her to go to the doctor and get medication for it, antibiotics for it. Therefore, the bacteria keeps on spreading. Right? It keeps on spreading. It starts in probably in the urethra, or maybe in the vulvar region of the vagina. So it starts spreading, and it travels up through the vaginal canal, all the way up into the cervix and uterus. And at that point, it will trigger localized inflammation, whether it's in the vagina, the uterus, fallopian tubes, the ovaries, or maybe throughout the entire reproductive system of the female. So again, that's called pelvic inflammatory disease. This bacterium can also infect other organs within the reproductive system, to include the prostate, right, to cause proctitis. It can cause pharyngitis, right, so this is infection of the pharynx. And it can also cause gingivitis, which is inflammation of the gums. Right? Newborns can also be infected with this, and for the most part, newborn babies that are born infected with it will show corneal infections, but they can also show respiratory tract infections. Okay, so in diagnosing the disease, right? It's pretty hard in asymptomatic patients. So if the patient does suspect that they are infected with gonorrhea, and they don't have any symptoms, then they're gonna have to get genetic probing. In patients that do show symptoms, so they go ahead and manage to get samples from the infected area, obviously from the genitals. So they carry out a gram stain, and they're looking for the gram-negative diplococci in pus samples of the patient. The treatment seems to be complicated because of the fact that many strains are resistant to antibiotics, so therefore they'll prescribe broad-spectrum cephalosporins, and these are administered intramuscularly, so in muscles, injected in muscles, and preventing, evidently, sexual abstinence, monogamy, limiting, the amount of sexual partners that that somebody might have, and then proper condom use, right? Also has, it can also be effective to a certain extent. Eye infections in babies can be prevented using antimicrobial. Okay, so meningitis is another disease caused by gram-negative bacteria under the genus Neisseria. It is caused by Neisseria meningitis, and it is very common among people that are under 20 years of age. So even though the bacterium can be normal microbiota, it can be spread out, and given that the person is immunocompromised, then this person can obtain it, right? They can get the bacterium by respiratory droplets, so by coughing, right? And people that are close to you breathe it in, and they can become sick with the bacterium. So this bacterium is pretty deadly, right? So it can cause death within 6 hours of the onset of symptoms. And it can move into the blood, causing septicemia, right? Which can evidently be deadly. Yeah, so septicemia is infection of the tissues and blood, and we're not supposed to have any bacteria in the blood, right? Normally we don't. So this bacterium can percolate right into the blood. One of the mechanisms of action of bacteria is the release of endotoxin lipid A. So therefore, causing septicemia. So we're looking at here in these diagrams is meningococcal septicemia. These are called petechiae, right? So these are evidence of the toxins that are released by this bacteria once this bacteria makes it into the blood, right? Again, lipid A endotoxin is released from this bacteria, and if it does manage to be percolated into the tissues, then it's going to accumulate in the tissues, causing tissue death, right? So the death of cells, which you're looking at here, right? Black tissue usually means that the tissue is necrotic. So this bacteria, this toxin, is basically killing the tissue there in this baby, in this person. Again, these are petechiae, which are manifested in patients with septicemia. In cases of meningitis, the meninges of the brain, right? These are layers. These are superficial layers, about 3 to 4 layers of the brain, 3 to 5 layers of the brain that protect the brain, and they circulate cerebrospinal fluid throughout the brain tissue. So this particular region is susceptible to infection by Neisseria meningitidis, and therefore, if the bacteria does make it into the meninges, then rapid intervention, right, is required. So the patient will show symptoms right away, and it's important that the person makes it to the emergency room, right, so that they can be treated, diagnosed and treated rapidly. So what are you looking for, right, if you are diagnosing a patient with meningitis or septicemia? You are looking for the gram-negative diplococci, right? And you are getting the sample from mostly cerebrospinal fluid. Although you can also test, these these tissues for the toxin. So you can get a sample from the tissue and test for the toxin. Treatment involves intravenous administration of penicillin. Intravenous tends to be the fastest way to get the antibiotic over to the tissues, right? If you make the person ingest, assuming that the person is aware, right, is conscious, then it's going to take hours, right, for the penicillin to take effect. Whereas if you administer it IV or intravenously, then this is pretty fast, because you basically deliver it to the blood directly, and as the blood circulates, right, it takes but a few minutes for the blood to circulate throughout the entire body. Therefore, taking that penicillin to the tissues really, really fast. Alright, so now on to the Enterobacteriaceae. Right, so just real quick, an overview of them before getting to disease is they are pathogenic, they're gram-negatives, and they are facultatively anaerobic bacilli, although they can also be coccobacilli, right? So these are very short rods, essentially, that look like coccus. Now, these bacteria generally live within our intestines, right, large intestines, thus being part of our microbiota. But again, any bacteria that multiplies in excess can be a pathogen. So these can act as pathogens as well. These are going to be ubiquitous in water, soil, and vegetation that is decaying. Ubiquitous meaning that they live throughout, right? So they're found in high quantities in again, water, soil, decaying vegetation, and again, these are enteric. Enteric meaning large intestine bacteria. So they're motile. They interact with nitrogen and glucose, right? In the fact that they reduce nitrates to nitrites, and they ferment glucose for ATP, right? So their metabolism again requires nitrogen, and they do use glucose as their source of energy, right? As most living things. So they do grow best in aerobic environments. They're not harmed by oxygen, but they're also anaerobes, right? So therefore, the facultative anaerobic nature of them. Some species, or these species rather, are distinguished based on their biochemical properties, right, of motility and the colonies that they form. Again, they are motile, meaning that they do move. They have flagella to be able to move. Pathogenicity characteristics of Enterobacteriaceae include membrane components, protein and polysaccharide capsular antigens, and these are used to distinguish among the different strains and species of the bacteria. So some of these factors that you're looking at here might be unique to some strains, but most of these would be found throughout all of the strains of the genus. So let's divide them up into antigens and virulence factors. What we're looking at here, if you recall, is a diagram of a bacterium. This is a generalized diagram of bacterium that contains a single cell. It contains a membrane. It contains a cell wall and a capsule. Right? There's DNA inside. That's the single chromosome of the bacterium. Here is a plasmid with the genes for their virulence and resistance. So if we divide these up into antigens and virulence factors, then we're looking at, for antigens, we're looking at outer membrane factors, right? So these are proteins, right, that are embedded onto the outer membrane, and they face the outside of the bacterium. And they use these to either attach to surfaces, attach to host, and they can use them as toxins and whatnot. One of the most important ones is lipid A, right? The endotoxin that I described in the beginning. There's a type 3 secretion system as well, which allows the bacterium to secrete substances from the inside. Keep in mind, right? This is DNA right here. DNA is divided up into genes. So gene fragments that contain a code are going to be expressed by the bacterium using ribosomes. So those ribosomes are going to go in and read the RNA that the bacteria made as a consequence of transcription of their genes, and therefore produce proteins. So these proteins are going to help the bacteria survive and thrive in a certain environment, and those are going to be secreted out through these systems here. Capsular K antigens, right? So if you recall from the immune system lecture, antigens are particular, moieties, if you will, particular domains outside of the found out on the membranes of bacteria and viruses, for that matter, right? So that's what antigens are, and they help them, invade a host and survive within a host. Capsular antigens, right, would be found on the capsule, right? They're embedded on the capsule of this bacterium here. There's also flagellar antigens, right? Flagellar meaning pertaining to the flagella itself that the bacteria uses to move. So those antigens are found right there in the flagella. And all of these can be used by the immune system to, present to dendritic cells, antigen-presenting cells, and they can be used as antigens so that the immune system can learn, right, using cell-mediated and antibody-mediated responses to be able to act against these infections. And in case of a future, right, because you always have a primary infection and then you have a secondary infection later on in the future. So if you do have antibodies against these antigens, then you can deter them in the future a lot easier than you did in the beginning. Virulence factors. Virulence meaning the pathogenic aspect of bacteria and their abilities, if you will, their skills that they do innately contain, that they do have, so that they can infect and cause disease in other organisms, right? They don't know that they're causing disease, obviously, but they to them it's basically just infecting tissues, right? They're infecting tissues. Why are they doing that? Well, because they need a source of nutrients, they need shelter, right? And they need to reproduce, and they happen to do so in tissues of humans and animals. So these are virulence factors that help them survive. Fimbriae would be one, right, for movement. Exotoxin would be another one. This is a secreted toxin that is used to deter attacks from other pathogens. Adhesin is an adherent molecule, right? Plasmids provide the bacteria with specialized genes to help them resist antibiotics, for example, and they help them also in their attachment and their infection processes. Iron-binding proteins is another virulence factor, and then hemolysin would be another one, right? And you can detect all of these. Hemolysin can be done quite easily in the lab by the use of blood agar, right? These bacteria are gonna go ahead and either partially or completely degrade the red blood cells within the blood agar using this toxin called hemolysin, right? So again, for more details on this and some of these antigens and virulence factors, go ahead and go back to the previous lecture so you can be more informed on these.
Enteric or intestinal pathogens possess a number of virulence factors. Two of these pathogens are Salmonella, the etiologic agent of salmonellosis, and typhoid, and Shigella, which causes shigellosis. Both bacteria are able to live within their host cells as parasites. Let's see how virulence factors help them to do so. Salmonella has flagella that help it to move in its environment. Proteins on these flagella inhibit phagocytes, as do other proteins in its capsule and cell wall. It also has fimbriae for adherence to the luminal surfaces of intestinal walls. It uses a specialized secretion system to inject proteins called invasins into the epithelial cell that cause rearrangement of the actin in its cytoskeleton. This rearrangement causes the membrane to ruffle, allowing the bacterium to be engulfed. Once Salmonella is engulfed by the intestinal cells, it remains in the vesicle, where it triggers an inflammatory response causing electrolytes and fluids to leave the cell. This results in secretory diarrhea. Inflammation also causes leukocytes to migrate to the region. When the bacteria are released from the epithelial cells, they either enter the bloodstream directly or by moving into phagocytes where they can grow. Shigella, unlike Salmonella, lacks flagella and cannot attach to the intestinal epithelial cells. They can only enter by attaching to M cells, which are specialized antigen sampling cells of the intestine. Shigella also uses a secretion system to inject molecules that cause the M cell to take it inside. Once Shigella is inside the M cell, it passes through to the other side within a vesicle. The secretion system has also released proteins that induce inflammation, bringing phagocytes to the area. Shigella can survive within the phagocytes by escaping the phagosome. The inflammatory response breaks apart the junctions between epithelial cells, allowing Shigella to spread. Another virulence factor of Shigella allows it to polymerize the actin molecules within the epithelial cells into a tail-like structure that propels it from one cell to another.
So diagnosis of these enteric bacteria does require of special culturing using McConkey agar, which you're familiar with. So in diagnosing it, once you've cultured, then you're gonna have to gram stain, right? So these are gram negatives. So what are you looking for? You're looking for a pink to red coloring, right, in their cell walls. Where are you gonna get your samples from? You can get them from the urine, from the blood, from cerebrospinal fluid as well, from feces, right? And aside from the gram stain, because the gram stain itself is not going to be enough, right? It'll help you differentiate between gram positive and gram negative, and from there you can decide, what route to take, right? But after that is you're gonna have to use several different biochemical tests, like for example the oxidase test or the hemolysis test, among many others, in order to be able to identify the bacteria that you're talking about here. For treatment, if it's enteric bacteria, then they can cause diarrhea, right? So that's one of the most common type of disease or disorders that these enteric bacteria cause, because they are found mostly within the large intestine. So with these, the treatment for diarrhea is basically just self-limiting, right? Ingesting lots of fluids, electrolytes, mostly water, and things like the Gatorade, for example, that contain the electrolytes lost to the diarrhea. So Gatorade is one, but they do sell specialized solutions, right? They do sell specialized fluids that are specifically dedicated, if you will, for people that are insufficient in electrolytes, for example, in people with diarrhea, right? So cleanliness, evidently, that would be another treatment measure there, because the bacteria is transferred over by feces, or transmitted by feces, right? So cleanliness is gonna be very important during the diarrheal manifestations there. So there's also antimicrobials that can treat the diarrhea or any internal infection. And last but not least is prevention. Again, as mentioned, cleanliness, personal hygiene, right? And evidently proper sewage controls. If you happen to be working in places unlike the United States, we have pretty good hygiene measures and cleanliness measures and sewage control for the most part throughout the United States. But if you happen to be working in places outside the United States, because of whatever reason, volunteering or whatever it is that you happen to be doing in your career, some third world countries don't necessarily have proper sewage controls. So something to keep in mind there. So on to the coliform opportunistic bacteria. So we're going to go ahead and divide up these into three main groups here: the coliforms, the non-coliforms, and the true pathogens within within this Enterobacteriaceae genus. We're looking at Klebsiella pneumoniae, which is one of the coliforms. The characteristics that differentiate these from the others is that these rapidly ferment lactose, right? So they're going to use lactose as a sugar for fermentation. And they do occur normally within microbiota, but as just like many others, they can be pathogens, opportunistic pathogens as well. The non-coliforms, they don't like fermenting lactose, so they use other means for fermentation. And within the coliform category here, we're looking at aerobic or facultatively anaerobic bacteria that are gram negatives, rot in shape, and again ferment lactose. This lactose fermentation does produce gas within a broth that is made up mostly of lactose. They are found ubiquitously in soil plants and also in decaying vegetation, right? They colonize the intestinal tracts of animals, evidently of humans as well. And the fact that you do find coliforms within water, right? So that's indicative of the impurities of water, and also of possible poor sewage treatment within the region there. So again, we're looking at Klebsiella pneumoniae here, which does cause pneumonia, right? A respiratory infection, but they're not, only exclusively respiratory here. These are also digestive, right? Which is indicated in the genus name, Entero. But again, they can also occur in the respiratory system, right? And they're actually pretty common, right? Pathogens of our lungs. They do contain the capsules that protect them from phagocytosis once they enter the body, right? One of the ways in which they fend off immune attacks is by protecting themselves, using their capsule to protect against phagocytosis. They do cause opportunistic infections, as mentioned, like for example, pneumonia rather. Right, so these can cause, again, respiratory and digestive infections, but they can also be involved in percolating all the way through to the blood, right, and causing bacteremia. They can cause meningitis as well, wound infections, and also urinary tract infections. E. coli is another bacterial species that causes opportunistic enteric infections, right? We do have E. coli as part of our normal microbiota, and we have worked with E. coli before, several different strains of E. coli, one of which you used in the transformation experiment, right? So that was engineered E. coli for the ability to take up the green fluorescent protein gene. So what we're looking at here is definitely not E. coli, right? This is Serratia marcescens. So you probably worked this before as well. You were probably one of the teams that were assigned as Serratia. So you notice, if you were assigned, you notice the beautiful coloration, right? That the colonies produce. So there is red colonies. So if you grow them on nutrient agar, which I believe it seems like this is nutrient agar right here. You grow them on nutrient agar, they produce these beautiful red colonies for you, right? So again, they are characteristic of the fact that they produce a red pigment when you grow them at either room temperature or all the way up to 37 degrees. I believe we grew them at 33, 34 degrees Celsius. But they will grow, all the way up to 37. Now, reason why these are healthcare, these have healthcare importance is, or healthcare setting importance, is because they do grow on catheters. So they're very common within catheters of patients, also within saline solutions, right, which is supposed to be, sterile. And they do tend to be found within hospital supplies, calling on the importance of autoclaving, right? All hospital supplies. You're going to go into the medical field, and maybe one or two of you guys are going to move on to working with autoclaving hospital supplies. So autoclaves are, these machines that work with pressure and high temperatures. So you insert, right, these medical supplies that are made up of metal or anything that can withstand the temperatures and pressures of an autoclave. And you set the autoclave at the right temperature, the right pressure, and it'll go ahead and kill everything for you, including Serratia marcescens. Yeah, so if left untreated, they can cause life-threatening infections. Left untreated, but also in more commonly in immunocompromised patients. They do show resistance to some antimicrobials. Other coliform opportunistic bacteria contain, there's a few. There's Enterobacter bacteria, Hafnia. There's Citrobacter bacteria. Let's move on to the non-coliform opportunistic Enterobacteria here. We are looking at Proteus mirabilis, right? Proteus mirabilis right here, very similar to Serratia marcescens, but these are facultatively anaerobic bacteria. So these are facultative anaerobes. These are most commonly associated with human disease of the urinary tracts, right? So they can cause urinary tract infections in patients specifically that have catheters for chronic amounts of time, if you will, so long periods of time. They can induce kidney stones, they can induce kidney infections, and they are resistant to many antimicrobial drugs. So other three non-coliform opportunistic bacteria include Morganella, Providencia, and Edwardsiella. These are healthcare-associated infections that primarily infect immunocompromised patients, patients with a low immune system. Yeah, and these can be, these can be patients with HIV, patients on chemotherapy, patients with cancer, right, or anything that basically forces a doctor to suppress the immune system of a patient, right? So these patients are going to be susceptible to infection with these types of bacteria. But for time's sake, let's go ahead and move on to the true pathogens, since we wouldn't necessarily have a lot of time to go into details on these three. Okay, so true pathogens of the Enterobacteriaceae genus include a few, right? Salmonella, Shigella, and Yersinia. The last one of which caused the fatal disease of the Black Death a couple centuries ago. So we're going to go and spend all of our time here talking about Salmonella, which we're looking at in this diagram. So what we're seeing is the Salmonella bacteria, right? Which is motile Peritrichus bacilli, right? Recall that. Motile means it can move. Peritrichus means that it contains a few flagella stemming from different locations, right, of their capsule, of their cell wall. And they're bacilli that live in the intestines of several different animals, right, and can cause infections in humans. Most human infections are due to the consumption of contaminated foods, foods with either human feces or samples that were brought from feces to the food, right? So go figure, right? Go figure how somebody could do that. Basically places that don't follow any sanitary standards. So poultry and eggs can also be sources of salmonella, right? And poultry and eggs that are either expired, right, or that have been killed within a few hours of selling it, and with no processing, no refrigeration, no freezing in between. This is very common in animal markets, right? Again, the reason why the virus that's infecting us right now, that's affecting us right now, SARS-CoV-2 came out, right? One of the main origins of it was from an animal market in China. So the meat from those animals is very fresh. This is very fresh meat. So those animals are killed right then and there, and they sell the meat within a few hours of having killed the animal. That right there is a cause for many, many different infections. I know SARS-CoV-2 is, and you know that it's a virus, obviously, but Salmonella can also be transferred via, live animal not live, freshly killed animal flesh. So two diseases of importance here are salmonellosis and typhoid fever. We're looking at the salmonella bacteria percolating into the blood, right? So what it needs to do is it needs to attack an excuse me, an epithelial cell. So once it attacks the epithelial cell, it enters, through endocytosis, which is caused by the capsule of the bacteria itself. So some virulence factors that we went over a few minutes ago allow the bacterium to endocytose, right? Cause the cell to endocytose the bacteria. Once inside, bacteria divides and it kills the cell, and in that manner, it manages to filter itself into the blood, causing, bacteremia, septicemia, also moving into other tissues, into other organs, and causing their disease there. So we're looking at the incidence of both typhoid fever and salmonellosis. We're looking at typhoid fever having come down, right, to very low levels from 1935 all the way, even before 1935, all the way to 2015, right? So close to 0 cases. It's a very rare disease here in the U.S. Salmonellosis has been on the rise ever since 1935, where we virtually saw no cases, right? To 1985, where we saw a peak, to 2015 right here, where we are basically keeping, right, almost at a plateau level right there. So typhoid fever is caused by Salmonella typhi, and humans turned out to be the only host for this bacterium. Carriers are often asymptomatic, meaning that they don't show any symptoms at all. And again, the bacteria are ingested by contaminated food or water, and it passes through the intestines to percolate into the bloodstream, right, where it causes most of the disorders, most of the problems with patients. Some of the problems can be gastroenteritis, right? So infection of this bacterium within the entire length of the large intestine, meaning the whole parts of the colon. For example, the ascending colon, the transverse colon, descending colon, the sigmoid colon, rectum, right? So it goes ahead and causes disease at any point within the large intestine. It can also cause bacteremia because of the fact that it does percolate to the blood, and lastly, it can cause peritonitis as well, which is inflammation and infection of the peritoneal cavity, which holds most of the the digestive organs. Treatment of these two diseases requires fluid and electrolyte replacement, since it is a large intestine disorder, infection rather, causing extreme diarrhea in both cases. So therefore, you're going to need to replenish the patient with ample amounts of fluid and electrolytes, as mentioned before. Typhoid fever is mostly treated with antimicrobials or antibiotics. Pasteurellacea is another genus of the gram-negative bacteria. These are small non-motile facultative anaerobes, so that require heme or cytochromes for growth. Heme is the functional group that contains an iron atom in the middle. If you recall, one of the cells that contains heme in your body are your erythrocytes, your red blood cells. They contain iron in them. Cytochromes are groups of proteins that are found mostly within mitochondria. So these cytochromes, and also in liver cells, in hepatocytes, right? So again, the fact that they are found in mitochondria, that essentially requires is indicative that they require NAD, right? NAD plus for their growth. NAD plus NADH is an electron carrier that we talked about back in metabolism when we were talking about glycolysis, cell respiration, Krebs cycle, chemiosmosis. Recall that NADH is one of the main electron carriers that is arguably one of the most important molecules in the human body and in the animal body for that matter, right? Because without NADH, then you cannot produce energy, right? NADH is that electron carrier that is required in its reduction to provide for those hydrogen ions that are necessary to provide the proton motive force during chemiosmosis. Yeah, so that would be in aerobic organisms. In anaerobic organisms like bacteria, for example, NADH is required for the cycling during fermentation. Yeah, so the way in which bacteria continue to produce ATP basically all the time, right, for their metabolic needs is by recycling NADH basically all the time, right? So these are going to require NAD plus for their growth. So they're going to go and metabolize NAD plus. There are two human pathogens of importance here. One is Pasteurella, the other one is Haemophilus. So we're looking at here in this diagram is Haemophilus influenza. Right? These like to colonize mucous membranes, particularly within the pulmonary system. So the respiratory system of animals can be colonized by this type of bacteria. It is an opportunistic bacteria, thus causing opportunistic infections in mostly in immunocompromised patients. Excuse me. Given the fact that they do like to infect mucous membranes, that means that they can spread throughout the entire upper and lower respiratory tracts of the animal. So if they do infect the lower respiratory tract, then they cause pneumonia, right? They can cause pneumonia, which can be deadly for an organism, for an animal. Other diseases that this bacteria can cause is conjunctivitis, sinusitis, dental abscesses, and also meningitis. Yeah, keep in mind, meningitis is not just left to one type of bacterium or virus for that matter. Is many different types, many different species of bacteria can cause meningitis, given that they do manage to percolate and infect the meninges, the meningeal layers of the brain. No, this can be prevented. There is a vaccine for these bacteria here, and the reason why they are so successful in infecting so many tissues of the body is their basically the fact that their capsule can resist phagocytosis. Another genus of gram-negative bacteria of healthcare and clinical significance is Bordetella. Right, so Bordetella are small aerobic non-motile coccobacilli, which are responsible for causing whooping cough. Yeah, so otherwise known as pertussis, I believe. Whooping cough is the common name. Pertussis would be the scientific name, right? So Bordetella pertussis is again the causative agent of whooping cough. This disease mostly affects children, but here we do there's a very low incidence of pertussis because of the fact that every single person here gets a vaccine against that. Now, this bacteria can become resistant to vaccines and antibiotics, and there's different strains of the bacteria that can cause the same disease here. Now, the reason why the bacteria is successful in infecting here is because of the following toxins. Right? It produces a few toxins here. First is the pertussis toxin, then is the adenylate cyclase toxin, thermonecrotic toxin, and the tracheal toxin. So the pertussis and tracheal cytotoxins, these infect mostly respiratory membranes. Yeah, so they help in adhering the bacteria on to the cells of the respiratory systems. Adenylate cyclase toxins basically disrupt the adenylate cyclase mechanism of any cell, right? So if you're familiar with that process there, that process plays a very important role in signal transduction of cells, as you hopefully saw in back in biology, right? The second stage of cell signaling is called signal transduction, which requires the use of these intracellular enzymes that are involved in the production of second messengers that have direct or indirect communication with the nucleus in allowing for cells to elicit responses to any signal that is incoming to the cell. So this bacterium disrupts the adenylate cyclase mechanism, thus preventing signal transduction within epithelial cells. So diagnosing this disease here, or this bacterium here, is essentially looking for the symptoms of whooping cough, which are basically usually diagnostic, and this cough lasts for weeks, right? So for more than 3 weeks. There is a prevention, right? And this prevention is a vaccine that is administered with immunization of the diphtheria, tetanus, and the attenuated pertussis or Tdap vaccine. So there are many more genus of the gram-negative classification of bacteria. I do highly encourage you guys to read a bit more on each one of the rest. For the sake of time, I'm going to go ahead and leave it right there. I covered some of the most important, clinically important, and healthcare important gram-negative bacteria in this lecture.